Hello, my name is Rebecca Janeta. I am going to talk you about Freedom Report Paternally Inherited IGF2 Mutation and Growth Restriction. Introduction. Half of the genetic material of each individuality it is of parental origin and the other half maternal. The genome inherited from the mother appears to be more important to fetal development, while inherited from the father is more important for the development of extra embryonic tissues. Application in the medicine. In the medicine is one of the epigenetic mechanisms involved in the regulation of gene expression in mammals. The mutant patterns are specific for each species and tissue type, forcing different regulatory proteins including DNA, methyltransferase, putative, deethylases, methylated IGF2 binding proteins.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letter A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called protease starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Pros and cons. Pros. One of the advantages of the epigenetic mechanism can be understood as a complex system to selectively use genetic information, activating and deactivating various functional genes. Epigenetic mutation can involve or cytosine residues in DNA and or change its chromatin structure that regulate gene expression. Cons. This process affects IGF2 protein interaction can alter DNA structure and replication, gene expression, and cell differentiation, cellular virus latency, and transcriptional. Makes this a topic of great medical relevance, where the general mechanics of DNA methylation and its with different pathologies in humans.
Alright, so as we wrap up section 5.3 of the Pierce text, we're going to talk a little bit about genomic imprinting, because this is vastly becoming a very, very intense area of research, especially in agriculture. So looking at the different ways that the genomics can actually be influenced by the environment that the individual is actually gestating in. So we see that there's a lot taking place in animal research. It's called fetal programming, to figure out exactly what is going on relative to each individual genotype. Alright, so genomic imprinting is defined as a differential expression of a gene that is dependent on whether it is inherited from mom or dad. So an example of this, as described in the book, is the insulin-like growth factor 2 gene, or IGF2. And this gene is responsible for directing nutrients through the placenta to the baby during gestation. So you can see that if there was a mutation in this gene, it would actually result in like premature babies or even miscarriages. So this gene is found on chromosome 11, and it's an autosomal gene, but it is actually determined based on which expression or which allele is being expressed, whether it was from mom or from dad. So relative to IGF2, the paternal copy is always going to be dominant and the maternal copy is silent. So the paternal genotype of IGF2 can determine the growth and gestation of these babies. So to demonstrate this has a really nice figure in your book. This is figure 5.18, And in the first segment here, we've got a paternal allele shown in red and a maternal allele shown in blue. And each of those alleles have different copies of the IGF2 allele. So as those copies come together, you're going to have very small hands and feet, be very short, and have severe mental retardation. They also tend to be heavier set or obese. And what ends up happening with these individuals is that they have a small piece of chromosome 5 that is actually missing. So it's a deletion, and that deletion is always inherited from the father. A second syndrome is Angelman syndrome, and this one we see usually in girls, but it's seen in boys too. And Angelman syndrome is where the individual has frequent laughter that can't be controlled. So they have infrequent and uncontrolled muscle movement. They tend to have a very large mouth, and they tend to seizure quite often. And this is also isolated to chromosome 5, and specifically a deletion on chromosome 5. And these individuals end up with this syndrome, and they will inherit it from the mother. So two different syndromes, both on chromosome 5, and very different phenotypes depending on whether they inherited the gene from mom or from dad. Some other syndromes that are really starting to trace back to being or the result of genomic imprinting include bipolar disease, carcinogenesis, and a slew of others. So there's about 25 total syndromes that have been isolated to genomic imprinting. All right. So as we finish up section 5.3, this really gives you a good breakdown of the different genetic phenomenon that we've been talking about, whether it was sex linked or genomic imprinting, and it gives you a description of how the phenotype is determined. So this is a good place to start studying.
Insue applied in Ecuador. Genetic evaluation of patients with postnatal hypogrowth in the molecular genetics laboratory during recent decades has allowed the identification of 65% neonates with molecular defects in practically represents one of the most frequent causes of postnatal. To establish and may vary depending on the diagnostic criteria and ethnic origin of the population and prevalence of at least children. In conclusion, it is predictable that implementation of new massive analysis techniques sequence and its probably repeats application to routine clinical diagnostic using the panel analysis of candidate genes exomes and her individual genomes at raising costs we allows within a few years to identify new genes and underlying remains relatively modest identified and molecular effects on the 10 to 20% of the cases studied. Thank you.